Hi everyone, this lesson is an introduction into the nervous system. Let's say you've just been bitten by a snake. Not good. Odds are it's probably going to end with you no longer being able to breathe. Pretty horrific, but I want us to think about this from a biological standpoint. Why is this happening? What is the neurotoxin doing to you chemically? What is happening that could stop your nervous system from allowing you to breathe? Well, thinking about the answer to this question is something that'll lead you to understanding how the nervous system works in its entirety. So why do animals have a nervous system to begin with? What's the whole point? Well, nervous systems are there to help organisms sense the world, to detect predators in the wild, and to control the body's functions. Odds are right now you're breathing, your heart's beating, you're digesting, but you're not consciously aware of any of it. Your nervous system is taking on that role. So how is this going to happen? With communication. The whole point of the nervous system is a way animals can send communication at a cellular level between the outside world and themselves and within themselves. To do this, they're going to use cells called neurons. You can see them on this slide. And these neurons are going to communicate to each other using molecules called neurotransmitters. For those of you that remember cell communication, this is a special form of paracrine communication. We're doing local communication. So if we're going to talk about the nervous system, we need to understand the special structures on a neuron. The biggest part of the neuron is called the cell body. In the cell body, that's where we'll find the nucleus, the organelles. This is the typical part of any animal cell. Attached to the cell body, you'll see these branching structures called dendrites. Dendrites are able to receive signals from other cells. They cannot send signals, but they can receive them and send them down to the cell body. The long tube-like structure running on the neuron is called an axon. This axon is just a super long fiber, and it's going to carry the message away from the cell body. If messages are received from the dendrite, that message is going to go through the cell body and then be carried down the axon. So our communication is going from right to left. At the end of the axon are axon terminals. These nodes are going to be able to send a signal to another neuron, and we call them axon terminals because they're located where the axon terminates. To return to the axon, you'll notice these blue circles covering it. This is the myelin sheath. The myelin sheath is fatty tissue that covers the axon. The whole point of this fatty tissue is it's going to speed up the rate at which signals can move across neurons. The myelin sheath is made by special cells called Schwann cells that continuously produce fatty tissue within themselves, making the sheath of myelin. That's why you see myelin represented as individual nodes, because each of those nodes is a Schwann cell. Now, what's the whole point of this myelin? Sure, it speeds things up, but why is that important? Well, if you have myelin on your neurons, the electric neural impulse can travel at about 150 meters per second. That's really fast. Without myelin, you're looking at only 5 meters per second. Pretty slow and sluggish. Reminder, we need a nervous system to detect the world and detect predators, so we need our system to be as fast as possible for the species to survive. You can actually see the effect of myelination and non-myelination on your own brain. Here with this cross-section of the brain, notice how some regions are white and some regions are gray. Why is this the case? Myelin is fat. Fat appears white. So the white matter in your brain is highly myelinated. It has a lot of myelin, so those are very, very fast communication signal pathways. Whereas the outer portion of your brain, the gray matter, has less myelin, so it appears gray and has a slower signal speed. For a neuron to communicate, it's going to use neurotransmitters, as I mentioned before, but it's also going to use an electric impulse. Here's the general pattern that neurons follow. They're going to go electric to chemical to electric. So starting with the neuron on the right, if a signal is received by the dendrites, an electric signal will move across the cell body, across the axon to the axon terminals, and that signal will then be converted into chemicals, into neurotransmitters. Those neurotransmitters can then be received by the dendrite of the neighboring cell, which will then cause it to switch back to being an electric signal. The gap between these neurons is called a synapse, and at that synapse, this is where the electric signal is going to facilitate vesicles opening and releasing neurotransmitters into the gap to continue communication from one cell to the next. To do this, I want you to think of it as dominoes falling. Understanding the action potential can get complicated real quick, so I don't want us to lose the forest for the trees. Think about someone who set up a series of dominoes. All of these have the potential to fall. The first thing you need to do is push that first domino. Once you push that first domino, the dominoes then continue to fall as a continuous wave. We say that the signal's being propagated, it's being carried through. Now, if I wanted to redo this, I can't when all the dominoes are fallen. So we're going to need to reset our system back up. We're going to need to reset the dominoes if we're going to be able to send signal after signal after signal. When a neuron's at rest, meaning it's not being stimulated, this is what the axon looks like. Notice that it's completely saturated in ions. I have sodium ions, Na+, I have potassium ions, K+, I have some chlorine, that's negative, and some amino acids that are negative as well. Typically, when there's no stimulation, there's a higher concentration of sodium on the outside and a higher concentration of potassium on the inside of the axon. If you've ever wondered why sports drinks like Gatorade are frequently promoted and you wonder what an electrolyte is, this is what electrolytes are. They're ions such as sodium and potassium and chlorine. Gatorade is actually nothing but flavored salt water. It was developed by people at the University of Florida to treat burn victims, and that's why it's called Gatorade. It was named after their mascot, the Gators. There are also ion channels on the axon. These ion channels are going to be able to regulate the movement of these ions in and out of the axon, and that movement is going to be primarily what's causing cells, such as neural cells, to be able to communicate. Because of this saturation in ions, axons actually have a charge. Notice how on the bottom I have a positive charge on the outside and a negative charge on the inside. Changes in these charges are going to be very important as we go through this. So at each step of a neural impulse, receiving a neurotransmitter, generating an electric impulse, and then sending a neurotransmitter at the other end, we're going to need to keep track of how charges change in the neuron and keep track of how these ion concentrations change, because it's the movement of this ion concentration that's going to cause the electric impulse to be generated, to propagate, and to move. So again, at rest, this is when a neuron is not being stimulated. You'll notice in the graph that it's at negative 70 millivolts. At this point, there's a higher concentration of sodium outside, higher concentration of potassium inside. That makes the outside positive, the inside negative. Why does the graph say negative 70 millivolts? This graph is measuring the inside of the axon. So at this point, it is more negative on the inside than the outside. Let's say our nerve is stimulated. So that means that you feel something, you taste something, you see something. When that neuron's stimulated by either external stimuli or by receiving a neurotransmitter, notice what's happened in the graph. We've hit what's called the threshold of excitation, also known as just the threshold. At this point, the charge inside the axon is now gone down to negative 55 millivolts. What's causing this change in charge inside the axon? Well, once it's stimulated enough, this is going to open the first ion channel. This is like pushing the first domino in our domino example. This is going to allow sodium ions to rush in through diffusion. No energy is being spent because I have more sodium on the outside at rest. When that sodium moves in, notice how it's changed the charge. We were originally negative inside, but now where the sodium's rushed in, it's positive. Because of this change in charge, this increase to positivity, we say that the neuron has become depolarized. Its polarization has changed. Now instead of being positive outside, negative inside, I am negative outside, positive inside. And this is what's causing that change that we see in the graph. This will start our electric signal. Now the first domino's been pushed, we've hit the threshold, so those dominoes will begin to fall down and the signal will be carried forward. Once that first domino's hit, the dominoes continues to fall. We continue to open ion channels and sodium continues to rush in. This propagates or carries the signal down the axon. Gate opens, sodium rushes in, we depolarize. That happens again and again and again in a wave formation. As that happens though, think back to the domino example. Once all my dominoes have fallen, I can no longer send a signal. That's a problem. Your neurons need to be able to be stimulated again and again and again. When we are depolarized, we can't re-trigger that. So to reset our depolarization, the neuron will then do repolarization. Here's how. Notice in the diagram how now gates for potassium have opened, and potassium is going out of the axon. When that potassium moves out, that's like setting the dominoes back up. We have now repolarized. If the opening of the sodium gates caused a depolarization, switching to positive inside, notice how the repolarization, the opening of the potassium gates, has now reset our charge. We're now back to positive on the outside, negative on the inside. And on our graph, you can see we've gone all the way up to positive 30 millivolts with the action potential as it propagated down the axon. But now with repolarization, we're going back down to around negative 70 millivolts. After this reset up, the neuron has a problem. At rest, we had more sodium on the outside, more potassium on the inside. But now we have the opposite. We've reset our charges through repolarization, but we need to reset our concentrations. We need to get sodium back out and potassium back in. To do that, the cell's going to use something called a sodium-potassium pump. Through active transport, so we're spending ATP, we're going to be able to push sodium back out and potassium back in. This requires a significant amount of ATP. This is why using your brain can make you tired. You need to be able to generate a lot of energy to use it. This also takes some time. It turns out that potassium gates close a little slower than sodium ones. So you'll notice in the graph that we actually go a little below negative 70 millivolts with something called the undershoot while we set up to return to negative 70 millivolts at rest so we can repeat this process again. Once it's complete, the neuron is back at rest so we can fire another signal. So let's review this all. So we started with resting potential. So let's review everything at once. We started at rest. At rest, we had negative 70 millivolts. We had more sodium on the outside, more potassium on the inside. Then something stimulated our neuron. We hit the threshold. Once the threshold was hit, that opened the first sodium gates, and we had depolarization. Our action potential, our electric impulse began to propagate across the neuron, and sodium gates opened sequentially. Potassium gates are still closed. Once we hit the peak, you can see that as number 4 in the graph, we then now have to set up our dominoes again. So we enter repolarization. The sodium gates close and the potassium gates open. With the reopening of the potassium gates, the neuron is repolarized. So the charge is reset. We go back to positive on the outside, negative on the inside, but our concentrations are off. So to reset our concentrations at the undershoot, we have the slow closing of gates and we have sodium-potassium pumps using ATP to pump these ions against their concentration gradients so we can set the dominoes back up and return to rest. So what's going to happen next? Well, what I just showed you is the electric impulse traveling along one neuron. Once that electric impulse hits one of the axon terminals, it's going to stimulate vesicles to fuse with the axon terminal and release neurotransmitters into the synapse, the gap between two neurons. Those neurotransmitters will then cross the gap and activate receptors. Those receptors then can be stimulated to hit the threshold in the next neuron, and the entire process can repeat again. Let's see an animation of all of this happening at once just for review.
The pain you experience when you burn your hand results so quickly in an action by your muscles. Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
To talk a little bit more about neurotransmitters, there are some common ones that are good to know. One really common neurotransmitter is acetylcholine. Acetylcholine is a neurotransmitter which, if received on the outside of muscle cells, causes muscles to move. Another is epinephrine. Epinephrine is often known as adrenaline. This causes the fight, flight, or freeze response if you're in a stressful or dramatic environment. And then there's serotonin. Serotonin is a molecule associated with having a pleasant or stable mood, your sleep, and your attention. I bring up serotonin because one of the most commonly prescribed drugs in the United States are SSRIs, or selective serotonin reuptake inhibitors. Here's how they work. And again, this is the most common antidepressant. What will happen when your neurons release serotonin into the synapse is it'll hang out in the synapse for a little while, and then the axon terminal will reabsorb that serotonin so it can be used again. As a human experiencing that, you experience mild happiness, and then it goes away rather rapidly. SSRIs, reuptake inhibitors, block the axon terminal's ability to reuptake that serotonin. So that causes more serotonin to be present in your synapses for longer than they normally would. And this tends to cause a stable or prolonged mood. Neurotransmitters can also be blocked. Looking at this example, at every moment your brain is sending signals to your lung to breathe. Your brain goes neuron to neuron to neuron, sends those neurotransmitters, that goes to your lungs. Right outside your lungs, there is a receptor for a neurotransmitter, And if that neurotransmitter is received, your diaphragm moves and you breathe. Let's say you're bit with a snake bite that has a neurotoxin. Here's how that neurotoxin works. It'll actually block the active site on the receptors in your diaphragm. With that active site blocked, that acetylcholine now cannot attach and cause your diaphragm to breathe. So even though your nervous system is sending signals, breathe, 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 those neurotransmitters bounce right off of the receptor. The message is never heard. The muscle is never stimulated to move. So we've gone through the anatomy of the neuron. We've seen now how a nerve impulse can be generated. We've gone through the phases of arresting potential and action potential and resetting the dominoes. And we've seen how different molecules like snake venom can interfere with the whole system. My hope is this was a helpful introduction to the nervous system, and I'll see you next time.